and welcome to this lesson for Advanced Higher Biology, which this time is on the cytoskeleton and cell division. So we're moving on from nerve impulses and communication to look at cell division and how it happens within the cell. And the first thing we need to consider is the role of the cytoskeleton within a cell and how it forms. So what you're expected to be able to do is describe the structure and function of the cytoskeleton in eukaryotic cells, describe the role of microtubules in cell division, and describe the processes which lead to the formation and the breakdown of these microtubules. So up until now, we've looked at basic eukaryotic cells and we've considered all of the organelles that are within them. And we've mentioned previously the cytoplasm. However, it's not just as simple as a jelly-like substance floating around. There is something else within there that we haven't mentioned up until now, and this is what we refer to as the cytoskeleton. The cytoskeleton is in fact a network of protein fibres that's extending through the cytoplasm in all eukaryotic cells. It's got a few different proteins to it. There are microfilaments, which are these ones here. We've got actin filaments down here, and we have microtubules. And the role of the cytoskeleton is primarily to give mechanical shape and support to the cell. So acts as scaffolding to help the cell maintain its shape. Remembering, of course, all cells are actually 3D structures. They're not the flat diagrams that we see drawn on when we look at diagrams. They are in fact 3D, and in order for them to maintain that shape and structure, they need to have this cytoskeleton within it. So we've got our three main protein fibres which make up the cytoskeleton. We've got microtubules, actin filaments, and intermediate filaments or microfilaments. Now, most of these will vary in different cells. So the sort of composition, the percentage of each type is going to vary from cell to cell depending on the sort of structure that an individual specialised cell requires. Each of these has a different role to play within the cytoskeleton. However, we're going to focus primarily on microtubules and not so much the other two, the actin filaments and the intermediate filaments. So organelles are also attached to the cytoskeleton, and it's got a role in controlling the movement of cellular components such as our vesicles and chloroplasts. Previously, when we talked about the use of vesicles to deposit useful proteins or proteins that are made within the cell, and we talked about them binding with the cell membrane, etc. We did discuss the fact that the secretion of those proteins, the movement of those vesicles, was controlled by microtubules, and this is essentially how that would happen. You would have the organelle, so a vesicle perhaps, attached to a separate protein. In this case, this protein is a kinesin protein, referred to as a motor protein, and the one that it's attaching to along here, this one down here, is your microtubule. So what the motor protein is essentially doing is it's made of a twisted fibre, and that fibre is attaching to the microtubule and walking the organelle along to its location, to where it's finally supposed to be going. The microtubule itself is a hollow straight cylinder composed of proteins called tubulins. So it's made up of alternating dimers of an alpha and a beta tubulin. So essentially a dimer is just something that's made up of our two different molecules here. So we've got our alpha tubulin here and our beta tubulin, and that's one dimer. So those dimers are going to combine to form this cylindrical structure, making our long hollow microtubule here. So forming a microtubule involves the polymerization growth of a microtubule. So polymerization just meaning we're putting small sections together to make a polymer. So we're adding all these dimers together to produce one polymer. So polymerization is the growth of the microtubule, and then we have depolymerization, which is a shrinking of the microtubule. And that's changing tubulin from polymerized to depolymerized. And when we do that, we're able to change the length of the microtubule, and therefore anything that is attached to that microtubule is able to move closer to or further away from the outer plasma membrane of the cell. And we'll look at this a bit more when we look at cell division specifically. Now, microtubules are found in all eukaryotic cells, and they originate from the microtubule organizing center, known as the MTOC, or the centrosome. So the centrosome, or MTOC, is found near the nucleus, and it contains centrioles, which are where microtubules are actually made within the centrosome. Microtubules can then play quite an important role in cell division because we need to remodel the cell's cytoskeleton in order to divide. So when a cell divides, we're going to have to recreate an entirely new cytoskeleton for both new daughter cells. And microtubules are also responsible for forming the spindle fibres, which are active during cell division. And they're the fibres that are involved in moving chromosomes from one side of the cell to the other and separating pairs of chromosomes into the two new daughter cells. So the image shows a cell which is dividing. We've got our microtubules here are shown in green. Chromosomes are the parts marked in blue. So from that, you should be able to describe the structure and function of the cytoskeleton in eukaryotic cells, being that the cytoskeleton is a network of protein fibres and that its role is to give mechanical support and shape to the cell. The role of microtubules in cell division is to help form the spindle fibres, which are going to move the chromosomes around, and also to help rebuild our cytoskeleton following cell division. And the processes that lead to the formation and breakdown of microtubules is the polymerization and depolymerization of tubulin, forming the alternating dimers of alpha and beta tubulin. As ever, if you have any questions, please feel free to ask as we go over it in class.